Hi, Mr. Lowell, and any other students watching. For our research project, we chose the topic of cloning, where we focused on both animals and humans. Our group was Jain, Mehta, Pandey, and Sreepathy. We chose to focus on the following part of the Star Wars clip.
They are totally obedient, taking any order without question. We modified their genetic structure to make them less independent than the original host.
What we derived from this clip was the following conclusion. Using genes and DNA receptors, it is possible to alter the behavior and traits of animals and humans. Through our research and findings of reading primary literature, we have determined that this is a fact, mostly. Our first experiment's objective was to compare the homozygous mutant and wild-type animals in an anxiety-related behavioral paradigm. This may sound complicated, but it is actually really simple. We did this by determining and counting the number of entries by a test animal into the open field and by measuring the time spent in the center of the open field. We used wild-type and receptor-deficient mice and measured the stress levels by sending them into an open field. The receptor-deficient males entered into the center approximately 3 times less frequently than the wild-type males, and they also spent 4 times less time in the center of the open field as compared with the wild-type males. Even when this data is normalized to the number of entries into the center of the open field, time spent in the center was still less in the mutant group. These results can also be seen in similar experiments, including an ESL clone experiment. And we can also see from these results that the receptor-deficient mice had greater effects in males as females as compared to males. The receptor-deficient females were statistically determined to enter into the center of the open field 2 times less frequently than the wild-type females. Our second experiment also had the same objective, but it was determined through a different manner. Instead, we measured the extent of anxiety by evaluating and measuring swimming and climbing activity of mice and their stress responses. The findings of this experiment were that with habituation, mice became progressively less mobile during the 6-minute test. By putting mice in what is called the Morris water bays, they had to run around until they found the hidden platform to escape the water. Instead of running around the circle, we saw that the less stressed mice would run through the center. The stressful nature of the forced swim is clearly shown by the activation of the hypothalamic pituitary adrenal axis, the article shows, and both receptor-deficient males and females were significantly more mobile as compared with wild-type controls. However, with this experiment, there were no gender differences.
In Experiment 3, the objective was to show the receptor audioradiography on the brain sections of wild type. This was done by measuring the level and density of the 5-HT1D sites on the brain in the wild type and in the 5-HT1B KO mice. In this experiment, we found that the density of 5-HT1D sites does not differ between the genotypes. In addition, we found that the more receptors did not result in the change of wild type caused by the presence of the 5-HT1B sites, which resulted in the disappearance of the labeling in the 5-HT1B KO mice. This showed that there was no significant effect on the genotype of the density of 5-HT1A binding sites, and there was also no effect of the interaction genotype cross in anatomical structure.
In Experiment 4, we went back to how new types of restriction enzymes could clone pieces of DNA as well as ligate them together, proving that DNA cloning is much more possible in the scientific world. So what happened is that for the experiment they used zinc finger nucleases, which are synthetic enzymes, took a chloroplast plasmid, which acts as a native parent DNA, so it's a naturally occurring plasmid and it's not synthetic, and they designed a specific target sequence for the plasmid and the zinc finger nuclease in order to cleave the long pieces of DNA as well as ligate them together. This proved that you can clone a DNA as well as ligate them, and it makes cloning organisms a possibility. However, it's not feasible because this is all done in vitro, one cell at a time, and it would be very hard to clone a whole organism. The findings do conclude, as said before, that these new types of restriction enzymes can clone DNA and make new plasmids from the plasmids already created, as well as be able to pinpoint specific target DNAs.
This article talked about how they targeted specific sequence of genes and engineered them using sequence-specific zinc finger nucleases followed by gene modification during subsequent repair. Basically, they used many of things called zinc finger proteins, and then they use methods like open, which uses bacteria to send bacteria to specific places and identifies them if they are feasible to use in this process. Other methods include two-finger modules and one-finger module. Structure and design of zinc finger nucleases is also identified in this article. Basically, cleavage domain of gene FOKI means that the gene to be edited, it needs an array of 6 zinc fingers. It also talked about gene disruption, which takes advantage of the errors introduced due to disruption or abolishes the function of a gene. In experiment 6, it was one of the contradictory examples. What it talks about was using stem cells. It talked about how to use stem cells and inhibit the growth of the gene. The one that's responsible for this is called DNMT1 gene, and how it works is it switches off the methyl group, which means that removing a methyl group from a gene sequence, and it inhibits it. The findings were that DNMT1 was the gene that was responsible for this activity. For zinc finger nucleases, it is a very specific thing. It can only edit things like a small sequence of DNA, but with stem cells, you are able to actually take a whole cell and clone it.
Overall, there were contradictions in experiment 3 and in experiment 6 where we saw that certain receptors had no effect, which was unremarkable. As we saw in experiment 1, 2, 4, and 5, our claim was mostly true. These contradictions can be reconciled through further research, especially in humans. However, we can state that the overall claim is still true.
Thank you.